Frontotemporal dementia. Frontotemporal dementia, FTD, also called frontotemporal degeneration disease or frontotemporal neurocognitive disorder. Brain frontotemporal dementia, FTD, is a group of brain disorders that cause slow damage to the frontal and temporal lobes of the brain. Busts in silhouette. It affects both men and women equally. Speaking head FTD usually shows up as changes in behavior or language, and the symptoms start gradually. Tear-off calendar. It most often appears between ages 45 and 65, but it can affect younger or older people too. Pill. There is no cure yet, and no officially approved treatments, but some medications and behavior therapies may help manage symptoms. Books. History and types of frontotemporal dementia. FTD was first described by Arnold Pick between 1892 and 1906, which is why it was once called Pick's disease. Brain. Today, the name, Pick's disease, is only used for a specific type of FTD where unique brain changes, called Pick bodies and Pick cells, are seen. These were described by Alois Alzheimer in 1911. Eyes. Common symptoms of FTD include major changes in personality and social behavior, lack of control, disinhibition, apathy and emotional flatness, problems with speaking or understanding language. Microscope. Each type of FTD is rare, and it mostly affects younger adults, called early onset. FTD happens due to a brain condition called frontotemporal lobar degeneration, FTLD, where brain cells in the frontal and temporal lobes slowly die, especially spindle neurons, over 70% loss, while other neurons may stay normal. Puzzle piece. Main subtypes of FTD. 1. Behavioral variant, BVFTD, once known as Pick's disease. 2. Semantic variant, primary progressive aphasia, SVPPA. 3. Nonfluent variant, primary progressive aphasia, NFVPPA. DNA. Rarer types include neuronal intermediate filament inclusion disease, NIFID, basophilic inclusion body disease, BIBD. Link. Some related conditions are corticobasal syndrome, CBS, CBD, FTD with ALS, amyotrophic lateral sclerosis. FTD usually starts between ages 45 and 65, but it can begin earlier or even later, in about 20 to 25% of cases. Busts in silhouette. It affects men and women equally. Bar graph. It is the most common type of dementia that starts in younger adults, and the second most common early onset dementia after Alzheimer's disease. Warning. The disease affects both thinking and behavior, and is recognized by health classifications as a cause of mental and behavioral disorders. Eyes. Typical signs include social withdrawal and disconnect from family, impulsive or compulsive behaviors, like uncontrolled shopping, inappropriate or vulgar speech, sudden screaming or emotional outbursts, loss of control over emotions, personality, and behavior. 3 o'clock. Symptoms usually begin gradually, and many patients may show behavior or language changes years before seeing a doctor. 1. Behavioral variant FTD, BVFTD, man standing speech balloon. Most common type. Major changes in behavior, personality, judgment. Signs: apathy, disinhibition, poor social manners, compulsive acts. Two. Semantic variant, primary progressive aphasia, SVPPA, books ? Loss of understanding for words and meaning. Speech sounds fluent but lacks meaning. Difficulty naming objects, people, or understanding word meanings. 3. Nonfluent variant, primary progressive aphasia, NFVPPA, speaking head hourglass. Speech becomes slow, effortful, and broken. Grammar and sentence structure decline. Comprehension remains mostly intact. 4. FTD with ALS, motor neuron disease, brain mechanical leg. Combination of FTD symptoms plus muscle weakness and wasting. Fast progression. 5. Corticobasal syndrome, CBS, CBD, hand with fingers splayed cyclone. Movement problems plus cognitive and behavioral changes. Stiffness, tremor, poor coordination, limb apraxia. 6. NIFID and BIBD, rare protein disorders, microscope. Very rare, diagnosed mostly on brain tissue after death. Show specific protein inclusions in neurons. Brain. Later stages of frontotemporal dementia, FTD. As FTD progresses, the different types can begin to overlap, and symptoms become more mixed. Fork and knife with plate. Many people with FTD develop binge eating and compulsive behaviors, craving sweets or carbs. Eating non-food objects, taking food from others. Brain scans, MRI, show that these eating habits are linked to damage in parts of the brain that control taste, reward, and self-control, like the insula, striatum, and orbitofrontal cortex. Puzzle piece. People with FTD also struggle with planning and decision-making, executive function, short-term memory, working memory, complex tasks like cooking, managing money, or organizing steps. Baby. As the disease gets worse, primitive reflexes, normally seen in babies, can return. Early sign: palmamental reflex, chin twitch when palm is stroked. Late signs: palmar grasp, gripping, and rooting reflex, turning head toward touch. DNA. In rare cases, FTD occurs with ALS, a serious motor neuron disease. Right finger. This combination usually means a faster decline and shorter survival. Droplet. Also, CSF leaks, leaking spinal fluid, can sometimes mimic FTD, but they are reversible if treated. Compared to Alzheimer's disease, more FTD cases seem to run in families, and researchers are discovering new genetic links all the time. Brain 1. Tau-related FTD, FTLD. Tau, caused by mutations in the MAPT gene on chromosome 17, which makes the tau protein. A condition called FTDP17 results from these mutations. Different tau mutations cause different types of brain damage. Some lead to PIC bodies, others to neurofibrillary tangles. Some affect glial cells as well. A rare tau-related condition called vacuolar tauopathy is caused by mutations in the VCP gene. Brain 2. TDP43-related FTD, FTLD-TDP, involves buildup of the TDP43 protein, often linked to several genes. GRN gene, also on chromosome 17, VCP gene, also involved in ALS and muscle disease. C9orf72 repeat expansion, one of the most common genetic causes. Rarely, TARDBP gene, which codes for TDP43 itself. Microscope 3. Other genes linked to FTD. CYLD, OPTN, SQSTM1, TBK1 involved in autophagy, cell cleanup process, TMEM106B. Certain variants, SNPs, increase the risk of developing FTD. Prohibited FUS pathology in FTD. So far, no genetic cause has been identified for FTD cases with FUS protein buildup. FTD isn't just one disease, it's a group of disorders with different protein pathologies and overlapping symptoms. Understanding the protein type can help guide research, diagnosis, and future treatments. Brain FTD and brain proteins after death, post-mortem findings. When doctors examine the brain after death in FTD cases, they often find clumps of abnormal proteins inside brain cells. These proteins are what define the underlying disease process, known as FTLD, frontotemporal lobar degeneration. Microscope. There are three main proteins involved. 1. Tau. 2. TDP43. 3. FUS. Together, they explain nearly all FTD cases. Bar graph. How common are they? 45 to 50% of cases, TDP43, FTLD-TDP, 40 to 45% of cases. Tau, FTLD-Tau, 5 to 10% of cases. FUS, FTLD-FUS, a small number have none of these, like cases with CHMP2B gene mutations. Brain FTLD-Tau subtypes. Protein clumps form different patterns. These include Pick's disease, CBD, corticobasal degeneration, PSP, progressive supranuclear palsy, AGD, argyrophilic brain disease, GGT, globular glial tauopathy, MAPT mutation pathology. NTD, neurofibrillary tangle disease. Brain FTLD-TDP subtypes. This used to be called FTLDU, ubiquitin positive, tau negative, type A, often linked to GRN mutations and some C9orf72 cases. Type B also includes C9orf72 cases. Type C, Type D, associated with VCP mutations. Type E, brain FTLD-FUS subtypes, rare, but include AFTLDU, atypical FTD with ubiquitin inclusions, NIFID, neuronal intermediate filament inclusion disease, BIBD, basophilic inclusion body disease. Magnifying glass in some rare cases, people who were thought to have frontotemporal dementia, FTD, during life were found, after death, to actually have Alzheimer's disease based on brain tissue analysis. Downward trend. The most severe brain shrinkage, atrophy, is usually seen in behavioral variant FTD, BVFTD, corticobasal degeneration, CBD. DNA. On the genetic side, a major cause of FTD is a repeat expansion in the C9orf72 gene. Other important genes include GRN and MAPT. DNA wrench. Recent research also shows that DNA damage and poor repair systems in the brain may be linked to the development of neurodegenerative diseases, including FTD. Warning: why it's difficult. FTD is hard to diagnose early because symptoms vary and often look like other conditions, like Alzheimer's, depression, or psychiatric issues. Test tube. Types of FTD symptoms. Doctors group FTD symptoms into three main types. 1. Behavioral variant FTD, BVFTD, changes in personality and behavior. 2. Semantic dementia, trouble understanding words. 3. Progressive nonfluent aphasia, PNFA, difficulty speaking clearly. These types can overlap as the disease spreads. Magnet imaging and brain scans. MRI scans may show brain shrinkage in the frontal or temporal lobes, but this can be missed early on. Scans over time can help show changes. PET scans help tell FTD apart from Alzheimer's, showing lower brain activity in different areas. Test tube CSF findings in FTD. Unlike Alzheimer's, FTD doesn't have a specific CSF biomarker that is universally diagnostic. But here's what we know. Microscope 1. Tau proteins. Total tau, T-tau, yellow circle usually normal or only mildly elevated in FTD. Red circle very high in Alzheimer's disease. Phosphorylated tau, P-tau, yellow circle normal in FTD. Red circle elevated in Alzheimer's disease. Checkmark. This helps differentiate FTD from Alzheimer's. Microscope 2. Amyloid beta 42, A beta 42, green circle often normal in FTD. Red circle decreased in Alzheimer's. Right finger so normal. A beta 42 levels in a patient with dementia suggest FTD over Alzheimer's. Microscope 3. Neurofilament light chain, NFL, measured by Simoa assay. Warning: elevated in FTD, especially in BVFTD and FTD MAND. Microscope reflects axonal degeneration, damage to long nerve fibers. Checkmark. NFL is emerging as a promising marker for disease progression and distinguishing FTD from psychiatric conditions or healthy aging. Microscope 4. TDP43 or FUS proteins. These are key pathological proteins in FTD, but they are not measurable in routine CSF tests yet. Research is ongoing to find TDP43-based biomarkers in CSF or blood.